Okay, we'll go ahead and get started. So this is the last lecture of this module, and your exam will open at 12:15. In zip grade, you'll have 24 hours to complete it. Please, I guess my advice, to get a separate sheet of paper, write your answers down, make sure that you're tracking them, enter them accordingly, and then submit when you're confident in your best effort. If you do run into issues, I check emails sporadically. Don't expect a response within like 5 minutes. If you're doing the exam at 3 a.m., I will be asleep, hopefully. But sometimes with the browser and internet connection, your responses can get kind of lost. I am able to track when you start and submit and your progress. So if you do run into an issue, please be truthful with me about what the issue is, because I'll be able to check that. But again, the best thing is to keep a separate sheet where you're recording your answers in case something does go wrong. We can work something out with that. So today what we're going to look at is another learning tool, another way of looking at molecules called the Fisher projection. And again, the molecule, all we're doing is changing our reference point. And the projection, as it's drawn, what we will see, it helps us figure some things out, but it really doesn't mimic the 3D shape of the molecule. So your own model kits will help you understand what we're doing today, because the drawings can get a bit confusing to interpret. So the Fisher projection is named after Fisher, who again won the Nobel Prize in chemistry for his work on sugars and purines. And so based on the number of slides today, I'm assuming that we'll have enough time. The stuff at the very end with carbohydrates is more of informational. It's not on this exam. But it will put into context why this projection is useful. And so carbohydrates are just another name for sugars, which you're all familiar with. And the main sugar for us is glucose. See this letter D here, this big D? That is not the same as the small d with optical rotation. As we'll see when we get further into a compound with multiple stereocenters, what exactly that means. And so this is the structure of glucose that Fisher figured out. And so let me put some numbers in here. So there's 6 carbon atoms. In total, 1 through 5, as drawn, are stereocenters. Carbon 1, however, it can racemize in a sense, but 2, 3, 4, and 5 are set, meaning that their absolute R and S configurations will never change. 1 can racemize, as I said, but that's more of organic 2 stuff that I'm not going to talk about. So if we use that rule for stereocenters, n stereocenters, 2 to the n, so 2 to the 4, that's 16 possible stereoisomers for glucose. So if we take 16 and divide that by 2, that gets us the pairs of enantiomers. So really what Fisher did was not only figure out the absolute configuration of each carbon, but which enantiomer that the naturally occurring glucose is. And so that's more of organic too in terms of the reactions and stuff and how he did that. But the reason that the Fisher projection is useful is if you're comparing 16 different things, how do you figure out the relationships between them? The chair is probably not the best way of doing that. So we're going to look at the carbon backbone from now a different viewpoint. And all we're doing is it's the same molecule. We're just going to kind of rotate things, flip it around, and look at it different. And the reason that's useful is when you're given structures, it helps you determine are they enantiomers, diastereomers, mesocompounds, or identical or different. What's the relationship? So as we normally draw things, I always just call that the zigzag drawing. I don't know if it has a formal name or not, but that's the carbon backbone that lies in the plane of the projection screen. And carbons, I want to use some colors here. So carbons 1 and 4, as they're drawn, are anti to one another. So typically in the zigzag, the carbon backbone has that anti-relationship. Or stated another way, the dihedral angle about the 2,3 bond leads to that 180 degree of the two blue labeled carbons. And so on carbons 2 and 3 here, I want to use the model kit to represent the Newman projection. So I'm holding the bottle like it's drawn. This carbon 1 in my left hand is, see how it's down, and then carbon 4 is up in that. And so for the Newman projection, all I'm going to do is change the viewpoint from the zigzag to this. So this would be carbon 1 now because it's down, carbon 4 because it's up. And you can see the red hydrogens and the blue hydrogens are on the same side. So my right, your left. And so that's the anti configuration or conformer of that. Now the Fisher projection, what we're going to do is orient myself. See how these blue hydrogens are coming out towards you? We're going to rotate around the central bond, that 2,3 bond, so that the methyl groups are now syn. So the dihedral angle is now at 0. So that's this center structure. And then notice the colors of the hydrogens. They're no longer on the same side. So in the anti or zigzag, they are, but when we do that, they're different. And so when we look at this Fisher projection, that's this last structure here. This is the eclipse conformation. And again, what I find tricky about the Fisher projection, we want this one down. This conformation is the most stable because you've reduced the steric interaction sections of the eclipse. Notice these carbons are far apart, but when I rotate it, they're now close together. And so here's the eclipse number. So to get to the Fisher projection, you have to do a couple operations. One of them is a rotation. And so when we did that, the red and blue switched sides. You're looking down a carbon-carbon bond axis in the Newman. To convert that into a Fisher, that middle thing, you're taking your Newman projection and then you're just rotating it this way. So that is a Fisher projection. As you see in the stereochemistry, the central bond, which I have as like a normal line, that's in the plane. The red-blue are projected out towards you, and then these methyl groups are going back. So those are the dashes. The stereochemical information is then reduced to just normal lines, the last structure of the Fisher projection. And that's what I want to be clear on so you're not confused. The things that are horizontal, the bonds, they are always projected out towards you. But again, they're not represented that way in the Fisher. For this 4-carbon molecule, this central bond is in the plane of the projection. But carbon 2 to carbon 1 going up, it looks like it's in the plane, but you see it's not. It's going back. So as you add more carbons to that backbone, notice how they're getting closer together. So if we sprout off even more, they're even closer together, even though in the Fisher, that vertical line looks like they're really far apart. So as we progress through this, and then you'll see for the carbohydrates, it's useful for stereochemistry, but again, it's not the best representation of the carbon backbone planes in which those carbons lie. On the vertical, they will always lie in the same plane, but notice how close they are together. And so when we see the actual Fisher projection for glucose, that vertical line is going to get pretty long, but what you have to realize is that those carbons at 1 and 6 are actually really close to each other, because they keep going back due to the eclipsing relationship. So we were proposing this from this point of view that looking at how do we know that the 2,3-dibromobutane does not form 4 stereoisomers, it's reduced to 3. And so let's draw or make one of them. And then on the next slide, we'll see why the other pair gets reduced to a mesopentane. So here we'll just use the blue ones for bromine. These red ones will be hydrogens. And so on the left structure, the 2R, 3R, I'm doing that rotation, and then I'm looking at it from a different perspective so we can see we have bromine and bromine. And then for the 2S, 3S enantiomer, you do the same thing. And then when you look at them, you can see their mirror images. So just to write some notes down here. Because each configuration of the stereo center switches to the opposite, those are enantiomers based on the nomenclature. And then you can see from the mirror plane sort of between them, that bromine is on the left, this one's on the right, right and left. So that's a mirror image. So structurally, those are enantiomers as well. Any questions on that pair? Yeah.
Those sigma bonds are rotation, like they can freely rotate. So in nature, everything would just go back to zigzag, right?
Yeah, so typically the anti-relationship is the favored conformer, or the more populated conformer. The Fisher projection where it's eclipsed or syn, it's just a human construct of how we can analyze these stereo centers in relation to one another. For 1 and 2 stereo centers along a backbone, it's kind of intuitive or straightforward. But as this backbone gets longer and you start adding more of these stereo centers, then you'll see the utility of the Fisher projection. That other stereoisomer, the 2R3S, this was the one where, in theory, there should have been 2 enantiomers, but they had the same name, which means they're the same compound. So when we do the same analysis for that, so this was the other, the things we just did. Notice that the bromines in the zigzag were on the same face. So now what I'm going to do is swap. Now they're on opposite faces. So that's what this compound is going to represent, this 2R3S. So when I do that rotation, now we have the eclipse or syn of those methyl groups. You see how the bromines are now on the same side in the Fisher? And so if we rotate, I'm going to rotate like this. So now that's the Fisher projection, bromines, hydrogens, and then your methyl groups. So the Fisher projection allows you to see a little bit easier that this is a meso compound, because the mirror plane is orthogonal to C2, C3. So the internal mirror plane, sort of the top, has to mirror the bottom exactly. So your bromines, your hydrogens, your methyls. And so meso compounds are defined as having a stereo center, usually multiple stereo centers, and then an internal mirror plane. So the optical rotation of these is going to be 0. So these compounds, if I did, so notice the way I'm holding it, the blues are the bromines. All I need to do, this is an allowed operation, is just taking this and rotating it that way. Notice I get the other one. So they're the same pattern. And again, the point of this is in the zigzag, the bromines are on opposite faces, right? When you do the rotation and look at it that way, they're on the same face. In a Fisher projection, what you're allowed to do is really only this. of the whole molecule. You are not allowed to do any sort of rotations of an individual carbon whatsoever. Because when you do that, you're going to change the configuration. So let me take some more notes here. So that's the internal mirror plane. So the bromines match, the hydrogens match, then the methyl groups match. So this red dot is an axis of rotation of the entire molecule. So maybe while I'm at it, I'm going to highlight one of these carbons in green. So you're not allowed to rotate one carbon because then again you're changing the configuration. So that sort of local thing is not allowed. And so again, the reason this is useful, it helps you determine the relationships amongst the different stereoisomers once you put them all in this form. Any questions? So we're going to look at another case related to this and how all of this stuff kind of evolved. And that relates to tartaric acid. And you can see the structure up there. And we can use this same model for that. So instead of methyl groups at the end, they're both carboxylic acids. But they're the same group, CO2H and CO2H. Then however we want to define it, whether the red ones are the OH groups and then the blue ones are the hydrogens. Kind of mimics what we just saw. We're just substituting some of the groups. So tartaric acid is 2,3-dihydroxy dibutanoic acid. And I think this gives a little bit more historical relevance to the evolution of, the stuff in your textbook. In 1769, it was isolated and extracted by Scheele from fruits and basically during the fermentation of or production of wine. So, throughout human history and trying to figure things out, what are we eating and drinking? And so tartaric acid played, I think, an important role in the development of the scientific method, but also spectroscopy. So that's the interaction of light with matter. And so earlier than that, in 1690, Huygens observed double refraction through transparent calcite. In 1811, Malou confirmed Huygens' result and described plane polarized light. And so within these last couple hundred years, people started to observe how, sunlight was interacting with these different substances. And then in 1832, Biot observed that tartaric acid rotated plane polarized light. So this is one of the first examples of polarimetry. And so that leads to these questions of why is this happening? And then in 1847, the famous Pasteur experiment using a microscope and tweezers was able to sort out these different crystals of this sodium. So sodium as one cation and then ammonium of the other. So of this compound, two different crystal forms were observed in terms of enantiomeric crystals, like not overlapping one another. And then that cascaded into how is this possible in terms of what is the molecular structure responsible for that. So how many stereoisomers are possible for tartaric acid? So we're going to use that asterisk to note the stereogenic center. So in theory, there's 4, but again, this looks like the system we just saw, right? So we know it can't be 4 because there's going to be that issue where the one pair has the same name, meaning that it's a meso compound. Yeah.
I thought you said that it did reflect plane polarized light. And if it does that, how can it be a meso?
So you're having one pair as enantiomeric, and then you have this meso compound, which is a diastereomer. So when the enantiomeric forms crystallize out, they're mirror images that will rotate plane polarized light. The meso compound will not. There is some ambiguity in the way this is written, but total there is, in theory, 4, but in reality there's 3.
So if this question was on the quiz, what would you want us to put theory for?
3. Again, this is a special scenario because your carbon 1 and 4 are the same thing, and your substituents on 2 and 3 are the same group. That's why it reduces from 4 to 3. But if you have these groups different, it can't be meso any longer. Is that? I would say 3.
So, is it possible to have more than 2 stereogenic centers where it's meso on all 3? So
Yeah, when we look at the higher carbon count carbohydrates, I'll point out the parents themselves are not meso, but reaction products of them can be. And we go into that more in 256, but that's part of the logic that Fisher used to prove the structure of glucose.
Okay, so these chiral centers only become achiral under eclipse circumstances?
So let me just correct that. the stereo center themselves, the configuration is set. So it doesn't change. The molecular chirality overall is meso due to the internal mirror plane.
Okay.
So when we go through the next couple slides, if that doesn't sort of satisfy your question, then we'll go over it again. So let's look at the structures where we're drawing. I think I have a mistake here. I do believe. No, this is good. So we have 2R, 3R. And then as it's drawn, the 2S, 3S. Those are enantiomer mirror images. Notice the carbon backbone is anti. And then notice the projections of the hydroxyl groups is drawn in both is towards the back. This other pair, notice, has the same name, 2R, 3S, 2R, 3S. And again, this is, as drawn, that enantiomeric pair. But what we'll see using the Fisher projection is that it's going to be reduced to a meso compound due to that internal mirror plane. And so from our initial model, really the only thing that we've changed is now we have acids at the end, and we've changed the bromines to OH groups. So again, what's challenging about converting the zigzag to the Fisher is that realization that the terminal carbons are folding back on each other. So maybe before I do that, if you can think about taking that center carbon-carbon bond. So the two carbons that are holding the OH groups. Then you do that rotation to the eclipsed form. Then you change your viewpoint to the Fisher. What happens with these OH groups in the Fisher projection? What's that? So in the zigzag, they're next to each other. When you do the rotation, they're going to end up on opposite sides. For this set, they're on opposite sides, but then when you do the rotation, they end up on the same side. And due to everything else, the H, the CO2H being the same, that's when they form the internal mirror plane. Methyls are the CO2H's, which I'm holding. The reds are the alcohols. And so looking down, there's your blue eyeball looking down the 2-3 bond. Which Newman projection do you think is correct? What do you think? So A. One of the things, even from just the way it's drawn without a model kit, the OH groups in the zigzag are on the same side. And remember that for the Newman, you're not rotating anything yet. So in the Newman projection, they have to be on the same side. So in my left hand, that's the CO2H group over here. See how it's down? So in A, that group is down. The carbon 4 is up on the back carbon. And then your OH groups are still on the same side. So that needs to be retained. So one second, I can't stress this enough. The model kits will definitely help you, train your brain patterns to doing these sort of rotations of objects and visualizing the molecules from different viewpoints. Yeah.
I was gonna ask, so in the previous molecular formula where VR was shown on the same side, it's like that half rectangle right now. But it was basically like there was VR on both sides of the molecule. It was going behind. How would we know if that molecule, if there was a Newman projection, it would be A? Because here it's on alternate. It's on top. The OH group is on top and bottom.
I guess kind of the way that when I think of this Newman, I see that the OH groups have to be on the same side. And then, you're looking at it, from this 90-degree perspective. But in my mind, when I'm taking my eye from here and then I'm moving it this way. And then when I do that, it's like my left eye looks at the stuff that's behind the board, and then my right eye, the stuff that's in front. But the other way of doing that is just making the model and then holding it. So you can see on my left side here are the OH groups. And from that viewpoint, they're in the back, the same side on the back. So here is a summary of that, where we have, for this set of enantiomers, notice the mirror plane is external to the stereoisomers. And then we're looking down the Newman projection in each way so that we can draw the enantiomers of those. And then this other one that ends up being meso, we do the same thing. So in the Newman as drawn, the OH groups are on the same side. As we convert it to the Fisher, we're going to rotate that bond, and you're going to see that OH group swing over. Over here, the OH groups are anti. So when you do the rotation, it's now going to end up on the same side in the Fisher. So those are the 4 Newman projections. The structures below are the rotation about the C2-C3 bond. So it looks like we're taking, in this case, the back carbon and then rotating it to this syn or eclipsing interaction. So that sets us up to look at the Fisher. So we're doing rotating this. So that's the Fisher projection here. And then in my mind, when I look at that Newman, instead of looking down this bond axis, my eyeball is coming up here and looking down 90 degrees to this bond, or like that. So the Fisher projection is just another frame of reference. The easiest thing, again, to do is take the zigzag, do whatever rotation you need, and then get it in this form, or the carbon backbone is then eclipsing. But you can see the OH groups are now on different sides, left and right of that Fisher. And then this is the Newman, and then I've done the rotation this way. So the take-home message is, from the zigzag, the OH groups are on the same side, and then into the Fisher, they're on opposite sides. So I talked about, that big D, big L, little d, little l nomenclature. The little letters correspond to the rotation of plane polarized light in polarimetry. The big letters are a stereochemical designation relating to, and now bear with me, I need to put some numbers in here. That's the carbon backbone. So carbon 1's always at the top. Then we have, in this case, 4 carbons. Let's just call that number N. If you take N minus 1 to arrive at the penultimate less than 1 stereocenter, if the main functional group lies to the left, it's L. Whether that means left or not, I don't know. If it lies to the right, it's D. So big L, big D correspond to the penultimate stereocenter. What side of the Fisher projection is the group lying?
Why would we start at the next carbon?
What?
Why would we start at the next carbon and not the first one?
So this nomenclature relates specifically to a stereogenic carbon. So carbon 4 is not stereogenic. You have to get back up into where your stereocenters are. And then you're, of that, it's typically some group and then H. Whatever that other group, what side it lies on, that's how you assign the L or the D. So naturally occurring carbohydrates on our planet Earth have the D configuration. And remember, that designator only corresponds to that one stereocenter. So when you eat food and there's glucose in it, it's going to be D-glucose. So again, the relationship as you see through the mirror plane in the center, those are enantiomers. You see the OH mirroring, the H's mirroring, these OH's mirror. and then these H's mirror one another. That other set, why it gets reduced from 4 to 3, is now we have that internal mirror plane. So when you convert the Newman into this Fisher, you can see that the external mirror plane is now internal. So for a meso compound, you have to have stereogenic centers with an internal mirror plane. That's the definition of meso. So the specific rotation of that will be 0. So you have one pair of enantiomers, and then the relationship of either of these to this compound is diastereomeric. So again, what does that mean? This and that will have different chemical and physical properties because they're diastereomers. And then let me just highlight this thing right here. So for the molecule itself, you're allowed to do a whole rotation of the molecule. You're not allowed to rotate any one center. So in the pair of enantiomers, you can see that the melting points are the same. The specific rotation has the same magnitude but opposite signs. So dextrorotatory. That's that little d. Levorotatory, that's the negative, that's the little L. So notice there's no correlation between that and that. The melting point of the diastereomer is about the same but not equal, and then the specific rotation 0.
So it just doesn't have a D or an L on it? It's just the...
Correct. So yeah, that brings up another point. So D and L, again, that's assigned to the penultimate stereogenic center. It has its own sort of subset of classification, but you can still assign R or S to that. Just using the normal CIP algorithm. Slides that I added. So anything from here on it's again not going to be on the exam. It's just more to put into context why this is useful. Any questions before we kind of move into the enjoyment part of the lecture? So we're going to start from this 3-carbon unit and then basically build up the carbon chain, and that allows us to see how carbohydrates are sort of synthesized either in nature or in the lab. This molecule in the center here is called glyceraldehyde. So it has 1, 2, 3 carbons in the backbone. There's only one horizontal line. So remember, a horizontal line is designated as a stereogenic carbon because you have the two groups coming out. And then again, this bond and that bond are going back because it's a tetrahedral carbon. So because the OH group is on the right, that gets the D designation. The optical rotation of this has a positive sign. So the enantiomer, the OH, is on the left, so that's L. And then the polarimetry measurement, that gives a negative rotation. And then this is showing you just what that horizontal line looks like. So those groups are coming out towards you. So we metabolize the D sugars. Most living organisms, I would say, do that, and that's interesting to think about because if there's a possibility of both the D and the L, why has nature chosen only one of them? Why can't we process both? So that gets down to the chirality of DNA and enzymes and basically how things fit in or interact with one another. So if you're into physics, we talked about spin. So spin can be clockwise or counterclockwise. The avalanche or butterfly effect of that spin on chirality and evolution is pretty interesting to think about. So in organic 2, we talk about amino acid synthesis. So amino acids have the L configuration. The amine group is on this side. And so that's what's used in enzymes and stuff like that. But why is that? Why have things evolved that way just based on the spin of an electron propagating through space and time? So that's a three carbon unit. So from this 3 carbon unit, what we're going to do is a reaction at the top carbon, and then we're going to add 1 more carbon. And so the chain's going to get longer. So we start with 1 stereocenter, that's the D and the L. From each of these, we can generate a set of children or progeny that's going to increase the number of those carbohydrates. We're starting with, on the left side, this D1. That stereocenter is set. We're going to do a reaction here that generates another stereogenic center. There's 2 possibilities. I think, yeah, these were the and we can think about carbohydrates as building up the carbon backbone and then adding a subsequent stereocenter each time. So let's look at the left-hand side first. So again, so that stereocenter was set because that's what we're building from. Then we're making a new one. There's 2 possibilities. Be on the left or it can be on the right. And then these are the L sugars. So we started from that L sugar. We now generated 2 more carbohydrates. So the pairs that you see here, the D, the L enantiomers, and then the D and the L enantiomer pairs. So what's the relationship between these 2? So these are diastereomers. You have one center that's the same, but the other one is different. So notice now in this case, the top carbon 1 and the bottom carbon 4 are different. So they're no longer the same. So now we can't have that meso. But say you took some conditions that reduced the aldehyde to an alcohol. So see how this is CH2OH? This would become CH2OH. The product of that would then be meso, because now there's an internal mirror plane. And so that was part of the logic that Fisher used, moving back and forth through these different series of compounds to figure out what glucose actually was. So we have 4 carbons. Let's do the same thing. We're going to elongate the chain. So see, for we're just going to focus on the D because they're the natural sugars. For this one, carbon 2 and carbon 3 are set. We're going to do a reaction at carbon 1 that's going to generate another stereogenic center. So it has 2 possibilities, meaning the OH group can go to the left or the right. So each one of those will do that. I like to think about it. Did I draw it? I kind of flipped it, but so from the 2 possibilities here, and the way that way. So in the 1D stereoisomer, see how it's right. Now you have these possibilities: right, left, right. And it's just combinations of that moving forward. So from this pair here, these sugars have weird names. I don't know the origin of them, but so from 4 to 5 carbons, notice these 2 stereo centers stay the same, and then carbon 2 changes. Again, these are just the D. The 4L enantiomers are not on the slide, but they would just be the mirror images of these things. Does the name of any one of these look familiar? So ribose. So deoxyribonucleic acid. So this 5 carbon sugar removes an alcohol, that's the deoxy part, and then it cyclizes into a ring. So the Fisher projection looks at the acyclic molecules, but remember I said as this backbone gets longer, these carbons start to close in on one another, and the vertical line is not a good representation of reality. So 1, 2, 3, 4, 5. This OH group will actually cyclize onto there to form a 5-membered ring. That's because that carbon and that carbon are really close to one another. They're not far away, which is kind of deceiving in the Fisher.
Can you reiterate the difference between capital DL and lowercase?
So let me do some drawing here. So right now we have 1, 2, 3, 4. In that example, carbon 3 is the penultimate stereocenter. The OH group is on the right, so that's given the D designation.
So that's just place on if it's right or left, and the other one is the rotation.
Yes. So notice how the carbon numbering changes. So see how this is carbon 4? It's promoted to carbon 5 because we're adding from the top. So carbon 3 in this case was the penultimate. Now it's carbon 4, but that doesn't change. That center does not change as you make these higher carbon count carbohydrates. So all of the ones on this slide are the D. So again, that was the 4 5-carbon D sugars. There's 4 of the L enantiomers. And then if you move into biochem or cellular and molecular biology, the 6-carbon count is pretty prevalent. Anything above that is probably not useful for undergraduate studies so much, but we're going to do the same thing for each of these. So each of these is going to produce 2 kids where that carbon stereo center can be left or right. So notice for the ribose, see how they're all on the right? We're going to add a group there. So that carbon becomes this carbon now. So we've put one to the right, we've put one to the left. Then we do the same thing for arabinose. So arabinose becomes glucose. And then each one produces 2. So shown on the slide are the D ones. The enantiomers are the L ones. And so that's the one we're consuming for energy. And I like to remember glucose as right, left, right, right when I do the Fisher. And so the tree is this. So the ones that are shown, really any relationship between them, they're diastereomers because you've changed one stereocenter consistently. The L ones would be the enantiomers. And getting back, to that question about optical rotation and things, like for allose. See how these are all on the same side? We can transform that group into CH2OH like that, and then that becomes meso. And then there's some other logic things that you can do chemically that convert a D sugar into an L sugar. I won't go too much into that. Maybe you can look at the structures, see if you can figure out what Fisher was thinking. But it has to do with symmetry operations. So you're looking for some initial symmetry present and then some chemical stuff to flip a D into an L configuration. So that was the last enjoyment slide of the lecture to really showcase why the Fisher is useful for determining stereochemical relationships along the backbone. So we'll end there unless there's any questions. I don't think it would be metabolized. I think it would be trying to pass through you. much about sugar metabolism, but you see that carbonyl group in aldehyde. Carbon 2 is alpha carbon, so that makes it acidic. You can depropionate it. Analyze and use the carbonyl acidity 2. From there, you can then form the other sugars. Maybe like isomerase. Maybe mannose gets isomerized with glucose in the body. But when you think about metabolism, human evolution has evolved it to use very specific energies. It doesn't want to spend energy metabolizing. Yeah. Yeah. I vaguely recall receiving an email a while ago. I'll have to go back.
She just wanted me to check it for you to make sure you got it. She's not gonna be back.
Okay. Yeah, I'll follow up with
Yes,
her.
sir. Thank you so much.